All right. Well, so thank you for the organizers for the chance to present. I'll just give a quick background of some of the work that we've been doing in the lab, speak briefly about the infectious clone that we use, and then get into our characterization of the cleavage mutant for SARS-CoV-2. So our lab, in collaboration with Pei-Young Shi's lab here at UTMB, designed a reverse genetic system. This is based on a system that was originally designed by Ralph Barrick's lab that disrupts the SARS-CoV-2 genome into 7 pieces. We can then grow these in plasmids. This disrupts a lot of the toxic elements and allows you to replicate the bacterial genome or the viral genome in the bacteria. Then we can extract those, sequentially ligate them, and then electroporate those into cells. And after that electroporation, we get full-length RNA. After the electroporation of the full-length RNA, we get recoverable virus that's equal to the wild-type virus. And so when we've done that, in the blue is our infectious clone-derived virus compared to the WA1 CDC isolate that was based on. And we don't see any appreciable difference in Vero cells. We've taken this into Calu-3 cells and other cell types and not seen any differences between the clinical isolate and the wild-type virus that's derived from the infectious clone. And so that's what I'll talk about today primarily. We've also generated a mutant virus that expresses a reporter gene, in this case M neon green, where we've placed the entire ORF7 locus. Previously with SARS-1, we had found that ORF7 was not a required accessory ORF. And so we have replaced it here with SARS-2 with this bright neon green derivative reporter virus. And so we can use that. We found that this virus also is not attenuated in an appreciable way in Vero cells, and you get nice bright green colonies in infected cells. We've developed a PRNT50 assay using this GFP neon green assay and finding good correlation with that. And I'll talk a little bit about that later and how we used it in these studies. But this has been a high throughput system that we've been using, and it's being tested using a lot of the vaccine platforms, whether it be Moderna or for the Pfizer trials. We've also developed a nanoluciferase assay, replacing that same locus with nanoluciferase. What we find is that this is not in as good a quality in terms of the PRNT50, the correlation analysis, but what it offers is rapid analysis. We can do this as quickly as 4 hours after infection. We can get a reading that's pretty accurate for our PRNT50s. And so this has implications for rapid testing of serological sera that might be used for convalescent plasma transfers, things like that. If you're particularly interested in any of these, you can get these reagents through the World Reference Center at UTMB. We have distributed the infectious clone pieces as well as the individual viruses, including the WA1, M neon green, and nanoluciferase viruses, as well as some of the mutants that I'll talk about today are going to be available all through the World Reference Center through regular MTAs. And so shifting gears and talking a little bit about the biology of SARS-CoV-2, everybody is now an expert in coronaviruses, so we all know how important the spike is in terms of its key role in coronavirus biology. It forms this trimer on the surface that the previous talk talked about in terms of this trimer that has these 3 parts. You have this top portion that's the receptor binding domain in red, and the NTD portion that's thought to have some role in interaction with the host cell. And then you have this, and that's kind of covered in this head attached portion. The second part is the stalk portion, which is highly conserved across coronaviruses. It contains the cleavage sites as well as the conserved fusion machinery. So for coronavirus entries, there's really 2 barriers for virus entry. One is the ability and capacity to bind receptor. In this case, for SARS-2 and SARS-1, the receptor is ACE2. We know that a number of bat viruses that are in the group 2B family do not bind ACE2. We've actually found previously there are bat viruses that existed in nature that had a capacity to bind receptor. But again, that's not the only process that's required for virus entry. The second is that a host protease needs to cleave the virus and allow its entry. In this case, this can be a serine protease at the surface like TMPRSS2, or it can be in the endosome through cathepsin-mediated entry. And so this is a 2-step process. You have to have the virus bind to the receptor like a key, and to turn that key, you have to have the protease activate the spike fusion machinery. Now we know for SARS-CoV versus SARS-CoV-2, The spike proteins that are greater than 75% conserved. We know from a number of studies that the ACE2 affinity is higher for SARS-CoV-2, which may play a role in its spread and transmission differences between the original epidemic strain. And importantly, for what we're talking about today, SARS-2 has a furin motif at its furin cleavage motif at its S1 cleavage site. And so this insertion site, this PRRA, is a canonical site that's recognized by furin for cleavage. It's directly upstream of the S1 cleavage site, and so it's thought to improve the efficiency of that S1 cleavage. So coronaviruses for furin activation and entry requires two cleavages, one at the S1 site, and then a second site at S2 that activates the fusion core activation pathways. And so this PRA is thought to make a major difference for this virus, and we'll talk about that in a minute. Now, just because there are furin sites in SARS-CoV-2 doesn't mean it's going to be more virulent. HKU1, MERS coronavirus, and OC43 are three coronaviruses that all have furin cleavage sites in their S1, upstream of their S1 site, but each of them has a very different pathogenicity and lethality rates. And so just because it has a furin site doesn't mean it, excuse me, going to be more lethal. And so what we were interested in knowing is, what is the impact of the furin cleavage site? What happens if you delete it and remove it? And how does that change infection, pathogenesis, and neutralization? And so the first thing we did was we modeled the structural sites of where this furin cleavage site was. This is work that was done by Kari Debink at Bowie State University, and she modeled it on the original SARS-CoV-1 spike. While Jason McClellan group and a number of groups like David Wiesler had generated a novel spike, the region that we were interested in was in an unordered region. And so we didn't have originally a shape or a location for this loop. And so Kari modeled it on this unordered loop that had some structure in SARS-1. What we found is that in blue is the wild type, and the darker blue is where the PRRA amino acids are. If you remove those, you shorten this loop to make this delta PRRA mutant virus. The important thing is you'll note that this is an exterior loop, and we don't anticipate that it has any effect on the overall structure of the rest of the spike, that everything else is somewhat maintained or predicted to be maintained relative to the original wild type strain. And so what we did is we deleted these 12 nucleotides to remove these 4 amino acids, and then recovered a virus that was able to replicate just as well as the wild type in Vero cells in terms of stock titer. When we look at the plaque assay, looking at the plaque morphology, we find that the furin deletion, dPRRA, that I'll talk about today, which will always be in blue, has a much larger plaque phenotype, about 3 times the size of the wild type. This is very consistent across every cell type that we plaqued it on, and very consistent that you can easily tell that this mutant has a much larger plaque size compared to the wild type. And so we wanted to go back and kind of take a look at what is different between these viruses moving forward, the mutant and the wild type. And so we started with Vero cells. This is the most common cell type that's been used to amplify SARS coronavirus. And what we found is that in general, the virus replicates with similar kinetics, but you'll note that at 24 hours post-infection, the delta PRRA has faster replication. It's about 25-fold increase in its viral titer relative to wild type. And this was notable because the PRA deletion, this deletion of this furin site, has been reported in a number of cell culture adaptation in both the CDC stock and the Munich stock. And so this is an issue for tissue culture adaptation, that the mutation that loses this furin site actually does seem to grow faster in Vero E6 cells. We've gone on to purify virions from Vero E6 cells and ask what the spike processing looks like. So coronaviruses can be processed before upon their release, likely by furin, but they maintain their spike structure, overall spike structure. With the structure, the spike already partially cleaved, you're already reducing the barriers for entry and fusion. And so we use SARS-1 as our control, and what you'll see is that the vast majority of the spike is in this full-length conformation, very little at the S1, S2 cleavage site, very little S2 prime. When we look at wild type SARS-CoV-2, we can see that ratio has shifted, that the majority of the spike is now at this S1, S2 cleavage product, much less full length. And here we've used N as our loading control for a nucleocapsid that's also on the particle. And then when you take that furin cleavage site out, now you can see in Vero cells, you've lost almost all of the S1, S2 cleavage product. Almost all of it is in this full-length form, really arguing that in the absence of that furin cleavage site, it really changes the processing of the SARS-CoV-2 spike compared to the wild type. When we quantitate this, you can kind of see this at the ratio here. Wild type SARS-1, really no processing of its spike. SARS-2, the vast majority, over 50%, is processed in the Vero E6 cells. And by taking out that furin site, you're losing the vast majority of that cleavage to that S1, S2 cleavage site. And so from this first experiment and analysis, we found that this PRA deletion has faster Vero replication in terms of cell culture adaptation. You can see why it would be selected for. It also has minimal spike processing. And so the loss of that furin site does make a difference in the actual virions processing upon its release. The next thing we did was we wanted to do it in a relevant cell type. In this case, we used Calu-3 cells, which is a respiratory cell line that's been used for SARS and influenza research over the past couple of years. And what we find is that the PRA deletion is attenuated in these cells. What's interesting is we don't see this attenuation over the first 24 hours, but at late time points, we have about a 10-fold difference at 48 and 72 hours post-infection, really arguing that the loss of this furin site does attenuate this virus, at least in this Calu-3, a respiratory line that's interferon competent. When we go back and we purify virions from these viruses, what we find is the same pattern, that SARS-1 has very little, almost all full length, very little processed to S1, S2. Interestingly, with wild type, and it's a little bit blurry here, we actually get even more processing to the S1, S2 site, arguing that the Calu-3 cells, these respiratory cells, actually process the virion more than what we see in Vero cells. And then with the PRA, we again see that there is a shift. There's much less S1, S2 cleavage site, but you'll note that there is a band visible here. When we quantitate that, you can see that ratio that we've increased the total amount of the S1, S2 cleavage site for SARS-2 in this particular cell type. And that even without that furin site, the delta PRA deletion still is getting processed more than SARS-CoV-1, arguing that there are sites outside of, or elements outside of this furin cleavage site that play a role in the processing of this virus. When we compare Veros to Calu-3 cells, we can see that there is cell type specific differences in how these cells are processed. You can see very clearly here with SARS-1 and the mutant that most of it's at full length with Vero cells, and then the S1, S2 cleavage site is absent in these two, but in the wild type. You can see with Calu-3 cells, we're getting a shift more towards that S1, S2 cleavage site, which may argue that there are differences based on the cell type and how these viruses are processed, and that the processing may be critically important in transmission and spread, depending on location. And we'll get a little bit more into that with some of the in vivo work. And so the results indicate this clear role for furin cleavage in SARS-CoV-2 processing of its spike, and that the processing varies from cell type to cell type. Depending on cell type, we also see differences in replication. The Calu-3 cells argue that the loss of the furin site makes the virus attenuated. In Vero cells, it's the opposite, where there seems to be a selective advantage. So we wanted to prove this fitness advantage in Vero cells and take a really close look at what's happening here. And so we did a competition assay comparing wild type to PRA at different ratios, 1:1, 10:1, 1:10. We then took RNA and examined by both RT-PCR and deep sequencing. And so this is a quick and dirty RT-PCR way to really look for this mutation. Again, we are worried that this mutation is a cell culture adaptation. And so this offers an inexpensive way to kind of look for this mutation that may contaminate stocks. And so basically you have these primers that are in, you have a forward primer upstream of the site, and then you have a primer in that PRA location, or generally upstream of it, and then you do a ratio comparing the two. And that'll give you an idea of how much of that deletion is in your population. About between 1 and 10%, you can kind of tell how much is there. So what we found is that when we put in the ratio at 1:1, we'd expect 50-50 on the top. And what we get is that the PRA deletion dominates, getting over 90% of the population when it's put in 1:1 after just one single round of replication in 24 hours. If we do it in the opposite direction, where we put 90% wild type to 10% PRRA, we can see again the PRRA deletion is dominant in these single round competitions. And obviously, if you do it in the inverse, that the PRRA is completely dominant when it's the majority of the population. That same trend is seen with the deep sequencing data with a little bit more clarity, but very clearly, the presence of the PRRA deletion in Vero E6 cells will quickly dominate and overcome the entire culture, making it a major mutation that you have to watch out for and control for. So the question is not really for cell culture adaptation. We know this mutation constantly appears. We've seen it in many of the clinical isolates after several passages of Vero cells. It's likely a when, not if. We do see elements of this as the virus is passaged. Less so in our infectious clone, but again, we're starting from a P0 pure stock. Obviously, this could have implications for all the SARS-CoV-2 research that's being done around the world, because obviously there's a difference in some of the readouts of these experiments. Now, what is the difference? And this is kind of something that we've just kind of fleshed out in the last couple of weeks. One of the major differences, obviously, Vero E6 cells and colony 3 cells, Vero cells lack interferon responses, so there's not a deficit there. But another aspect is that Vero E6 cells have very little to no expression of TMPRSS2, the serine protease that I talked about at the beginning, whereas colony 3 cells are known to have a high expression of TMPRSS2. And so we wondered if this was actually the difference in the fitness between the two cell types. And so to look at this, we took Vero cells on the left here. This is wild type that I showed earlier. And then we had Vero cells exogenously expressing TMPRSS2. And what we find is that when we overexpress TMPRSS2 on the surface of Vero cells, we remove the majority of that advantage for the furin deletion virus compared to the wild type. When we do the ratios again, 1-to-1, on the right panel, you can see that the TMPRSS2 Veros maintain roughly close to 50%, very little advantage. We think with deep sequencing that the PRRA mutant has a small advantage, but by overexpressing this exogenous TMPRSS2, we've actually removed that selection on Vero E6 cells, which has implications for propagation and use of this Vero cell line. Another important thing is when we grow these viruses on these TMPRSS2 cells, what we find is that the ratios are very similar to what we see off of regular Vero E6 cells. And so while there are small shifts, we'd argue that the virus is coming off of TMPRSS2 Vero cells, expressing Vero cells, is very similar to regular Vero E6 cells. And so the barrier is, one, not on exit. It's more than likely an entry issue that's distinguishing the two viruses and the cell types. It also means that these TMPRSS2 Vero cells may be the ideal cell type to replicate and propagate SARS-CoV-2 for many studies. So in summary, for the in vitro stuff, use of the TMPRSS2 removes that advantage for the mutant. It may be really important in cell culture adaptation questions. Virion processing is similar between Vero E6 cells, and so we could argue that use of either of those could be relevant and isn't expected to shift the expected results. But really, the important effort is that we have to make sure that we at least know what is happening with this mutation in these stocks when these studies are done. Without that, we can definitely expect that some of the results could be skewed, and I'll talk about that here in a moment. Now, shifting gears, the in vitro stuff is useful, and the attenuation in respiratory cells was novel, but we wanted to take this a step further and look at in vivo experiments. And so with this mixed results, we decided to test these using the hamster model, and this is work that we did in collaboration with Alex Freiberg's lab here at UTMB. We took two groups of 4 male hamsters and infected with either 10 to the 5th plaque-forming units of wild type or the PRRA deletion virus, and then took weight loss, disease score, oral swabs, and nasal washes over the course of 28 days. And what we find is that the wild type virus causes a very consistent 15% weight loss, peaking about day 8. And the PRRA deletion actually causes no weight loss whatsoever, causing really clear attenuation of this mutant in the hamster model. When we look at disease score, we find that the wild type mice, or the hamsters, have augmented disease between day 6 and 10, peaking at day 8. And again, we didn't see any signs of disease. And our disease signs are ruffled fur, hunching, uh, significant weight loss, or loss of movement are the metrics that we use. And so again, the PRA deletion virus causes very little to no disease that we measured in this hamster model. When we look at the viral titers, what we find is something that's a little bit surprising in that we don't see attenuation in titer at the nasal washes or oral swabs. If anything, at some locations and some time points, there's actually higher replication, although some of those differences are relatively marginal. It's not as if this mutant is attenuated because it doesn't replicate. It's that there's a change in the amount of disease that it causes. We subsequently took these animals, which all recovered their weight by 28 days, and re-challenged them with wild-type virus to see if they were protected from subsequent re-challenge. And so what we find is in blue and red, animals that got the PRA mutant or the wild-type mutant were completely protected from subsequent re-challenge, whereas the mock obviously lost that 15% of their body weight, as we saw previously. So again, this argues that the PRA deletion not only doesn't cause disease on the front end, it also protects animals from further challenge with SARS-CoV-2. We also noticed that there was no disease found in the PRA deletion. One of the wild-type animals did show some level of ruffled fur and hunching, but really mild disease compared to the original infection. Interestingly, when we looked at wild-type and PRA deletion vaccinated animals following the second challenge, the re-challenge, we did see measurable titers in the nasal washes and viral RNA, arguing that while there is a significant reduction in viral replication, it's not completely sterilizing immunity in these upper airways, arguing that, and this is consistent with what's been seen with mice, and may signify that upper airway and nasal pathways may not achieve the level of immunity that's completely sterilizing. Although it's not clear that these viruses are really transmissible or infectious in terms of transmission purposes moving forward. So for the hamster data, the loss of the furan site clearly attenuates the SARS-CoV-2, but it's not a replication attenuation. We know that the furan deletion protects from subsequent re-challenge, and that we haven't achieved the sterilizing immunity that we may have hoped for. Now, all of this is useful in that it tells us that the virus is attenuated, at least in the upper airway and in terms of its disease causing, but it doesn't really explain what's happening in the lung. And so to evaluate what's happening in the lung, we partnered with Mike Diamond and Adam Bailey at Washington University. They had access to the K18 human ACE2 expressing mice. And so what they did is they infected those mice with wild type or the Delta PRA mutant. And what they found is that there's attenuated disease in terms of weight loss. Again, the PRA deletion causes less weight loss, less disease. What they found was that there was actually less replication in the lung at early time points, although at late time points, those do equilibrate and they're very similar at later time points. Interestingly, when they did FlexEvent, which looks at lung mechanics and lung respiratory function, they found that the wild type had much more damage and loss of respiratory function relative to the PRA mutant, which stays much closer to the mock. And when they looked at the histopathology, what they find is that, again, there's not much disease. Yes, sir? Oh, sorry. They found that there's not much disease in the wild type. In the wild type, there's a lot of disease, and there's less in the PRA. And so this obviously has implications for vaccines and moving forward, but it's clear that the mutation drives attenuation in this disease model, in two different in vivo models. So the last thing I'll talk about is neutralization. So the PRA mutant may contaminate stocks, and so we're worried, what does this mean for vaccines therapeutic studies? Does this shift the PRNT50? And so to do these studies, we generated a GFP expressing, M neon green expressing gene, expressing virus with the mutant on the y-axis here and the wild type on the x-axis. And what we find is that when you have the PRA deletion virus, you see a shift in PRNT50 values about 2 to 3-fold. You require more serum to neutralize that virus. And that's true whether it's the high sera here on the right, intermediate, or low sera. Very consistently, you need more sera to neutralize this mutant than you do wild type. And so what the question was, why? And so it could be a change in spike conformation, although we didn't think that was likely. We know that the spike processing could expose wild type sites that are more cryptic, or alternatively, there just may be less spike on the surface of the wild type virus because of the processing. And so to look at this, we use monoclonal antibodies against the RBD and find that same trend that you need more antibody to neutralize the virus off of the PRRA deletion than the wild type, arguing that there's actually more physical spikes that need to be neutralized on the wild type, on the mutant virus, than the wild type. And so why is that shift? Well, we had one theory, but we wanted to go back and look at EM and kind of really take a closer look at these virions. And what we find is that in the left, wild type virus, we can easily see particles, and they were generally in individual states. What's interesting is that when we looked at the PRA deletion, we'd actually find these clumps of virus. And when we focus in on these clumps, you get these clumps of 3 to 6 viruses, really driving up the level of virions that need to be neutralized. We also noticed that the PRA deletion has a much higher particle to PFU ratio, which really indicates that there's just more particles that have to be neutralized. And that's true even if you control for each clump being a single particle forming unit. And that gets to a ratio that's close to that 2 to 3-fold difference that we're seeing. It's interesting that about a third of the particles in the PRRA deletion are in these clumps, really arguing that they have this big clump of virus that may be much more difficult to neutralize. And we don't really have a reason yet on why that clumping is happening, although it's very consistent across many of the stocks that we've looked at and generated. And so this PRA mutant has this higher particle to PFU ratios, probably driving its difference in its neutralization profile. And it partially explains that. The important thing is that this neutralization profile is going to be different. And while it trends to argue that the neutralization will be in the direction of less neutralizing, we have to really consider that when we're comparing vaccines and therapeutics, this mutation can really shift very much what could be selected moving forward. And so it's a key element that we have to look for contaminations. And so in conclusion, we know that the furin site plays a key role in fitness and virulence of the virus, and that this culture adaptation in the SARS-CoV-2 backbone has to be considered for all SARS-CoV-2 studies moving forward. We also know that based on some of the cleavage stuff, that there's sites outside of SARS-CoV-2 that increase that cleavage. So it may not just be the furin site that's augmenting this transmission and spread of this virus. We're in the process of doing a number of more in vivo studies and EM, but for the most part, these are studies that we're hoping to finish up in the next couple of days or weeks. It's been under review for a little bit now. With that, I'd like to thank my group. Brian, Kumari, and Craig have been doing the heavy lifting on the SARS-2 work here at UTMB. Nathan Bopp did all the EM work. Pei-Young Shi's lab, obviously with the help with the reverse genetics and a number of the PRRT assays. Alex Freiberg did the hamster work. Adam Bailey and Mike Diamond did the mouse work at WashU. Kari did the modeling. We, of course, have funding from the NIH, and all of these reagents are available through the World Reference Center at UTMB.
Thank you, Vineet. Let's see if there are questions. Please raise your hands. So while additional people think about potential question, I have one. So you're measuring essentially the effect of the virus on the hamster based on the weight loss, but most of the morbidity and comorbidity, mortality and morbidity of the virus is associated with its inflammatory sort of cascade of events. And I was wondering whether you have been able to measure any, for instance, of cytokine releases, et cetera, following infection with the dPRRA, because obviously that could still potentially cause some undesired effects in the host.
Yeah, so so that's the mouse work we just finished the last couple of weeks. So we have cytokine data from mice showing that there's less immune stimulation in the mice that are infected with the PRA. Not that they're absent, it's that it's just 2 to 3-fold less protein, cytokines, chemokines. When we look at the infiltrate and the immune pathology, and kind of had to go through it quickly, it's just we get these focal areas of immune pathology in the PRA deletion, but they're very focal as opposed to it's widespread in the wild type. And so we think that it's just inducing a much less robust immune response and less immune damage and inflammation.
I see Steve has a question.
Hi, Vineet. So this is Steve Goff. Beautiful talk. Just the kind of things that I think need to be done. So I was curious about when you think the cleavages are happening that are assayed in the virion. So when you're looking at the virion particles, the cleavages must have happened on the, as I think of it, as on the way out,
Yes.
as the cell is producing the virus. And I wonder, is that in the Golgi or ER? And that must be a guess where it's happening or on the surface. And then, of course, then the virions on the way in, are subject to cleavage again.
Yeah.
So it's like when does it, and I guess these different mutants are happening at different times, but maybe one of those guys is happening on the way out, the other guy is happening on the way in.
Yeah, so my bet is the S1 cleavage is happening on the way out. So this gets into hand wavy stuff, and I see Tony on here, so I mean maybe Tony can maybe comment on this too. The spike particle is thought to keep it, even after S1 cleavage, the spike is thought to keep its trimer conformation. And it's really receptor binding that's giving access to that S2 site. And so you have that S1 cleavage. If it's already, once receptor is engaged and S1 is cut, that top head can kind of cleave off or be removed, and then you have access to that S2 site. And so what I think happens is when on that release, you're getting this S2, the S1 is getting cleaved on the way out, but it's maintaining. But this gets into a question that we're seeing, and one of the things we think is happening is that there's a lot more cell-to-cell fusion with the furin cleavage site mutant versus the wild type. And so one of our theories is once it engages that receptor, it can easily fuse because it's going to get cleaved very easily because that access to that S2 site is there. Without that cleavage site, that spread may be different because it's not easily getting cleaved. It's got to go through that two-step process.
Right. So it's sort of predisposed, ready to go, I guess.
It's primed in a way, and it gets to a question, and evolutionarily, in the lung, it might be okay for that to happen. But in a bat gut, that may be a killer in the digestive tract. And so that may be something selected for against in the bat viruses. So it may be just something that can work in the respiratory tract and maybe not work as well in the digestive tract.
Right. So roughly we're thinking S1 on the way out, and then S2 on the way in. Yeah. And the S1 cleavage might make it easier for the S2 to happen on the way in, but only in some settings. And so so there are analogous things with HIV.
Yeah. Well, I think in location-wise, I think the TMPRSS2 expression is much higher in the airways than it is deep in the lung. And so I think that gets into a little bit, I think the efficiency of the replication between different SARS-1 and SARS-2 in the lung may be about that TMPRSS2 activity. We have data from SARS-1 that really argues that it's not efficient with TMPRSS2, and that SARS-2 is much more efficient with it, and it may be because it's already partially cut.
I mean, it's perfect. It's great that you have already this situation where the mutant in one cell line is good, and in the other it's bad. So it's clear that it can be good or it can be bad, depending on the cell line. Anyway, nice, thanks.
Yeah. Thank you, Vineet. So I don't see any additional questions. So I think
I had a quick one actually. So in your cells, your tempers to Vero cells, I don't know, did you show the actual titers coming out of those cells? Were they like the same, or did that reduce titers in the Vero cells? You showed the ratio evened out between.
Yeah, so the endpoint titers are the same. Now that gets into, we purchased the Vero cells, so we didn't add them to our Vero cells, and how Vero cells are from cell to cell. In terms of pure titers, they're the same. I do think that the cells are a little bit different from each other.